My name is Gruber, and I'm an assistant clinical professor of clinical pathology here at NCSU.
I am Alexander Tufano, and I am a lab technician underneath Dr. Gruber.
I'm Adriana Villasenor. I'm a third-year veterinary student here at NC State.
The Gruber Lab is fundamentally a veterinary comparative immunology program. So my background is in clinical pathology, and then my research background really focused on macrophage biology and understanding the function and regulation of macrophages. So when I started my program here, I was really interested in understanding how macrophages are reprogrammed in various environments, and a really important environment is the tumor environment. So we know that tumors will take control of the macrophages, which has a number of outcomes and promotes tumor survival. So the macrophages downregulate immune regulation and suppression of the tumor, but it also macrophages actually promote tumor cell growth and metastasis. So I was really interested in how do tumors reprogram macrophages, and then what are the consequences of that for tumor biology. So we have this really exciting project that kicked off last year investigating canine osteosarcoma, which is a primary bone tumor in dogs, primarily affects large breed dogs like Great Danes and Labrador Retrievers, but looking at the interactions between macrophages and osteosarcoma cells. I had the good fortune to have veterinary summer program students last summer, and one of those, Adriana, did a really fantastic foundational work on this project. So I'm going to let Adriana talk about that.
Yeah, so like she said, I got involved in the Gruber Lab through VSP offered by NC State, and my project was looking at how supernatants from osteosarcoma cells grown in culture would affect the gene expression of macrophages. The basis of the work that I did here was we cultured osteosarcoma cells in media, and then we used that media containing cytokines or other mediators and used that to stimulate the macrophages. And then we ran the macrophages through qPCR to look at classic pro and anti-inflammatory gene expression changes. And we found that the macrophages, when stimulated with the supernatant from the osteosarcoma cells, induced a more pro-inflammatory phenotype in the macrophages.
And what was really interesting about that is that the two of the genes that Adriana looked at were TNF alpha and CXCL10, which are both cytokines that are pro-inflammatory but have also been associated in human osteosarcomas with a worse prognosis, higher metastatic disease. And so that work, and this was a pretty significant change in gene expression by the macrophages, so all that we had from Adriana's great work was that the osteosarcoma cells were secreting something. And so that something is what we're investigating more with this grant that we just recently received from the American Kennel Club Canine Health Foundation. And for this grant, we're going to be now narrowing down what are those signals that the osteosarcoma cells are secreting. Are they cytokines? Are they other soluble factors? Are there some insoluble factors? So that's part one of that grant. And then part two is now we know that macrophages will secrete increased amounts of these pro-inflammatory cytokines. So what effect do those cytokines have back on the osteosarcoma cells? So we're really looking at it from two angles: the effect of osteosarcoma cells on the macrophages and the effect of the macrophages on the osteosarcoma cells. And that's where Alex has started investigating some of the classic cytokine signals that may be mediating that, and is going to be pushing that project forward in the next 6 months, 9 months, along with hopefully another student or two.